Okay, so it gives me great pleasure to welcome Andreas to virtual Oxford and CompChem Kitchen. We are about to hear in the next 30 minutes or so all about applications of machine learning, artificial intelligence in drug design. What is realistic? What are illusions? How can we stop kidding ourselves? So take it away, Andreas.
Sure. Great. Yeah, great. Thanks a lot, Garrett, and everyone in Oxford for the invitation to speak here. So what I will talk about is recent developments in AI and drug design. And to evaluate that a little bit critically, there are many things happening, of course, and there are many valuable things happening as well. But let's look at that again and what can actually be useful in the future when we look at the data we have in chemistry and biology, different types of applications. So first of all, I would say this is an academic talk. So I'm working both at AstraZeneca as well as the University of Cambridge. So today I'm speaking purely in my capacity as an academic. So everything presented is my own personal opinion and not that of any employer, funder, or anyone else. What is the situation of AI and drug discovery today? There are many things happening, and so I think it's time for critical evaluation of where we are. So here are some headlines and screenshots collected from the internet recently. And it's great, in my opinion, that there are many resources going into the area because that really enables us to try new things and to apply algorithms to analyze chemical and biological data and to really validate things, even compounds going into the clinic, for example, and to really validate new approaches. But on the other hand, we're all getting older, and I'm certainly getting older as well. And so I'm old enough to remember, for example, the 2000 biotech bubble, where I was working on a cheminformatics startup. Actually, that's where I got in touch with the field. That was also the time of the Human Genome Project, for example. And so sometimes things turn out to look a bit different a few years later. So I went back through to some of the papers that were published around that time. So Trends in Biotech in 2001, for example, Thomas Reis said, the number of drug targets will increase by at least 1 order of magnitude, and target validation will become a high throughput process. Or for example, there will be more drug targets, 3,000 to 10,000 targets, compared with, at that time, 483. But recent 2017 estimates of drug targets put the number of targets that are targeted by drugs on the market at around 667. That's not to say that nothing happened. Of course, we have new modalities, for example, and also the Human Genome Project was really instrumental in ordering and sorting the information we have. Organizing genes, for example, we are now able to enumerate the genes we have. We're able to catalog better what we have and to map genomes, for example. But still, at that time, around 2001, obviously there were many expectations, and not all of them became true years later. And so this is, I think right now, there were so many things happening in AI and drug discovery for a number of years already. So much funding was going into the area that it's time to look at what does the field look like right now and to evaluate that a bit more critically. So what I will talk about is the data landscape. I will touch on, well, easy labels, easy models. So where can we actually build models, AI or machine learning models? I will touch on deep learning, of course, talk a bit about biology as well, which I think is a bit neglected in the field right now, and also talk about the interface of AI, human psychology, as well as society. So this is a slide I quite often used in order to start my talks, in order to introduce what we do in our group. So we're looking at molecular structure, so chemical structure, which is reasonably well defined, depends on stereochemistry or tautomers, for example, but still, by and large, we can store chemical structures reasonably well. Then we have some kind of protein or mode of action level, and we have some type of phenotypic information, and we also have some links between those. So we have bioactivity data from Chem or PubChem. We have something we could call pathways, but of course that's more human concept. The whole idea of a pathway is not necessarily the way biology thinks. And we also have some type of phenotypic response data. And so this looks quite neat. And I would call it, for the time being, the world is flat model. So here we have everything, all our information neatly organized and labeled, and we can make links between those data sources. And if we can do that, that's of course great. And that was the motivation for the work we did in our group, because we can answer practically relevant questions. For example, the question, what is the reason upon treatment with a compound for phenotypic effect, so for mode of action? Or we could ask the question, which compound should we make to target a particular pathway, so particular proteins in a pathway, so we can map back to chemistry. Or we can even apply that to personalized medicine. We can sequence, for example, HIV, that's reasonably cheap, doesn't cost much at all. And then we can say which patient responds better to a certain drug treatment if we analyze the sequence of HIV protease or other enzymes of that virus. But unfortunately, the world is not flat. And so if we think back to that triangle of mapping compounds to targets, as well as to phenotypes or effects, that is unfortunately not as black and white as we would like it to be. So links between drugs and targets and disease are quantitative and often incompletely characterized. So for example, when we think about drug-target interactions, there are subtle differences in compounds effects. So we can have partial or full agonists, we can have different downstream signaling, for example, different residence times, and all of that influences compound action. So it's not that a ligand acts on a target, it's much more subtle what that compound does. And also effects are state dependent, of course. So we have variation between individuals, and that goes far beyond genome differences. So even if we sequence patients, that does not tell us everything that's going on in that person. So we have lots of variation between individuals. Drug absorption, for example, is influenced by what you eat in the morning, or your diet, or your lifestyle, or whether you're smoking, and so on. And often that's not captured in the data we have. And in particular, phenotyping is particularly difficult. It's very subjective. Of course, deep phenotyping was coming up recently, and that might be an answer or lead us to more populated data resources in the future. And of course, if we're trying to integrate all of that, we don't properly understand biology. So we don't even know what to measure and what to label. And quite often what we measure in biology is, of course, more technology push than a scientific pull. So we measure what we can measure because there are new techniques around. But that doesn't mean that that's the right data type we need in order to understand the biological system. I will come back to that later. So basically the point is this neat triangle we had before, that's a very idealistic view. In reality, things don't look like that. So what should we do now? And if we think about data and the data we have available and how we can use that, that depends hugely on the context. And you can organize it by stage of drug discovery if you wish. So we have early discovery stages and later stages, or safety related stages. So in early discovery, what you have quite often is you have simple readouts. So you, for example, measure bioactivity against the isolated protein. So you have Ki, Kd data or IC50 values. And that means two things. So on the one hand, you have a large number of data points, which is good for training models. And models, and this is also good, data points have clear labels. And so that's also good for model training. So you have relatively homogeneous data. So you can say these compounds are active on a particular protein. Sometimes you mix up different types of data, so IC50 or KD data and so on. But still you have relatively homogeneous data and you have lots of data. And so in this setting, it's relatively easy to train models. So that's good because you have many data points and they're clearly labeled, but it's probably less good for in vivo relevance. Yeah. On the other hand, if we look into later stages or into the safety context, we have more often quantitative data, and effects depend on dose or exposure, for example, and labels become more fuzzy. So classes depend, for example, on dose, on exposure. We also looked at histopathology data, for example, and you don't have single labels. Whether you have particular events in the liver, for example, depends on a set of endpoints that you need to consider simultaneously. So you have conditional labels and you have sets of labels that don't make sense individually. You need to look at them in combination. So what is the situation here? You have less and less clearly labeled data. And that's much more difficult from the machine learning angle. So you have smaller data sets and you cannot assign clear labels. And in addition, even creating the data and storing the data is tricky. So if you think about animal studies, for example, it depends on where you put the cage in a particular room that influence your readouts. You have events also in background, so untreated animals and so on. So later stage data, safety data is much more difficult to deal with. And you can really put particular data you have in one category, so it's early stage, large numbers of data points, or into a later stage context, we have much more quantitative data that doesn't have clear labels and is much more difficult to deal with. And it's a very different setting for machine learning then. Obviously, the early discovery setting, it's much easier to apply machine learning algorithms, you use artificial intelligence. If you have more complex data, that's much more tricky, and in some cases, I would say even impossible. So this was the data, and of course also the problem setting is different in early discovery versus late stage or safety. So in early discovery, quite often you're in a discovery setting. So the question is, out of many possible options, many molecules you could make, out of 1 million or 10 to the 63 or so, give me some that are good enough. And so anything fulfilling a set of criteria will do. So you're predicting presence of something, and you can use generative models in many cases. On the other hand, if you're in a later stage or you think about safety, you need to make a prediction for a particular data point. And here you have a large number of criteria you want to rule out, different types of safety events. And you are asked to do that based on limited data. So you're trying to predict the absence of many things. And in this case, you need predictive models for a particular molecule that you're interested in. Yeah. And so this early stage setting, that's easier because you have many possible solutions, and anything will do that fulfills a set of limited criteria. And in the late stage context, you're forced to make a prediction for a particular data point. And you have a much larger number of criteria that depend on quantitative measures. So it's much more difficult to analyze data in that context. But where can we use early stage data? So this is an area we worked on quite a lot in the group. So sometimes you have a link between a molecular structure and a phenotype, so an effect. You have a cytotoxic compound or you see an adverse drug reaction. And what you can do with target prediction models is you can predict a protein target based on data or hypothesize what might be a protein target. And that protein target might then be responsible, provided the compound reaches that target at a sufficient concentration. In practice, which you quite often don't know, then this target might be responsible for a particular effect. And so we did so actually for the last 10 years or so. We also published some public target prediction models, for example, of Lewis Mervyn when he was a PhD student with me. And so all of that is published in the public domain, so it's accessible to everyone. So we can hypothesize based on millions of data points we know what a possible protein target for a ligand could be. And this uses data that we have, and I think that can be very valuable, and that clearly belongs to this first category I mentioned of having early stage data, large amounts of data with relatively homogeneous labels. So what I would say about this area is that using bioactivity data for ligand protein activity modeling is relatively possible. I say relatively possible because it's limited by the data we have. It's difficult to deal with data biases to make, for example, absolute predictions of the likelihood of binding to a target. Imagine you have one chemical series or one activity class where you have 10 very homogeneous compounds, similar scaffold, one scaffold. In another class, you have 10,000 very diverse ligands. And then you're trying to normalize how likely is it that you're binding to a particular target if you have so different data. That's tricky. But still, generally, in my opinion, it's possible to model bioactivity data for simple events, ligand-protein interactions, for example. So that is possible. It only covers known chemical space, of course, but at least you know the data you have, which is good. It suffers from various data biases. Normalizing model output is not trivial. And of course, what you need to look at afterwards, is that relevant in vivo? So for example, is your protein actually expressed in the organ you're interested in? And you need to consider quantitative aspects afterwards. Still, we make use of bioactivity data we have, which is certainly a positive thing. A while ago, I started to think about which areas do people actually focus on when thinking about AI and drug discovery. And so this is a slide that people will certainly not agree on, but it's trying to illustrate something that I think is probably generally true right now. So on the x-axis, what you see is, different areas, research areas in AI and drug discovery. On the x-axis, you see the attention that the field has received in the recent past. In my opinion, subjective judgment, of course. So maybe in the last 3, 4, 5 years. So not in the last decades, only in the last few years. On the y-axis, you see whether the scientific question is solved, but of course, in quotation marks, you can always argue at what stage a question is solved, but progress towards reaching the goal, you could say. In blue, you have chemical aspects, in green, biological ones. And the size of the circle is what I would describe as the impact of bringing a drug to a patient. Historically, 20, 30 years ago, the drug discovery was quite driven by chemistry, and that changed maybe 20 years ago. So biology became much more important. And so chemistry is more the early stages. How do I synthesize a compound? Is it active against my protein? And then you go into later safety assessments, for example, currently in many cases still into animals, quite often early into humans, of course. So, and these are more the biological stages. And so what I think is generally true is that much of the AI and drug discovery approaches focus on the chemical stages. And that can be a reaction prediction. That was quite an area of intense research recently, target prediction, what we also worked on. And all of those areas are very important, and they are areas where we have lots of data, but they're quite far away from the bringing a drug to the patient. So there's hardly any analysis of biological complex late stage data, but I would argue if compounds fail quite often due to insufficient efficacy or toxicity that you see only in humans, and maybe that's an area we should increase our focus on. So we deal basically with the areas where we have data. So AI goes where the data is. So maybe we look for the keys simply where the light is. Maybe we need to shift our attention a bit to the more biological stages of analyzing data. And there's one reason for that, and that reason is biological data is painful, of course, and that is difficult to analyze in many cases. So for a data scientist, you might ask the question, so does a drug now cause an adverse drug reaction or not? But then you ask your pharmacovigilance department, and you get the answer, if you have a patient with this genotype and this particular disease endotype who takes the drug, but only at that dose, and so on, then we see an adverse drug reaction. But of course, not in every case. And adverse drug reaction can be more or less severe, for example, as well. So we cannot label this type of biological data so easily. And much of the information we need is actually unknown. And that is very difficult to handle. But that is the typical case in biological data we have. So this is an example from human data. But analogous, of course, that's true for cellular systems. So we have cell line drift. We have media matter. For example, we were working on antibiotics. So whether you see resistance to a particular antibiotic depends on how you grow your bacteria in many cases. If you have animal histopathology data, it matters where you put your cage. Is the handler male or female? That increases stress in the animals. And so you get different responses. And so those complex systems, they're really difficult to handle. And that makes it difficult. And so I guess many people shy away from those types of readouts. Let's go to the area of deep learning. So there are three brief examples I would like to mention here. So deep learning certainly can work well, in my opinion, not only my opinion, many other people have shown that. Sometimes it works well numerically, but it doesn't really address the underlying question. And sometimes it's pushed, in my opinion, in a biased way in publications. So where can deep learning work well? For example, in target prediction. So there's a study from Andy Meyer's group at the University of Linz with Sepp Hochreiter. So he was comparing different target prediction methods, and you see on the plot, feed-forward neural networks performed on average best. So there are areas where you have relationships between the data. You have many data points, clearly labeled data, where this can perform quite well. But of course, in practice, what you need to take into account is trade-offs. So how long does it take you to optimize the model? If you need to update your model regularly, every 2 weeks, let's say, is that really worth doing? And I would also like to point you towards slides that Anthony Nichols was giving at the AI and Chemistry conference here in Cambridge last September. And he was giving a presentation with the title, "Is your machine learning telling you anything you didn't already know?" And so he's asking the question, is there really a significant difference between the performance of methods, and does that matter in practice? And I can definitely recommend having a look at those slides. They're on the website down here. And so there are areas where deep learning certainly can work well, in my opinion, and this is one of them. Lots of data clearly labeled. Here it makes sense. But then, and this is a paper we were working on before, sometimes, in my opinion, deep learning gives you good numbers, but the question is, does that really matter in practice? And so this is an example from modeling the synergy of anti-cancer compounds using deep learning. So recently, many public data sets became available by Merck, AstraZeneca, the NCI Almanac, and so on, where drugs were screened against cancer cell lines in combination. Those response matrices is quite a lot of work. And so we modeled those, and we were trying to predict synergy. which is sometimes desired, sometimes it's not because we want to capture escape mutations, but in some cases it's desired. So we were trying to model synergy of compound combinations. But I was asking the question, so we published that a year ago. I was a very good master's student as well. But afterwards, I was asking myself the question, so does that matter in the drug discovery context, in the real world, in the drug discovery context, not just as a machine learning exercise? So what we did is we used deep neural networks. So we input compound combinations, drug A, drug B, so combination of drugs. You need to have two representations both ways around to make it symmetric, and also genetic data, so gene expression data, for example, or other types of cell line information to model synergy. So we were trying to predict whether compounds have a synergistic effect in this cell line. And so you can evaluate that numerically. So you have a particular parameter you choose. And so we had a true positive rate of 0.55 to negative rate of 0.95. So basically that means 1 out of 2 positive predictions is correct, and 19 out of 20 non-synergistic predictions are also correct. So these are the numbers. And for a model that sounds reasonable, might be useful in practice. But is that actually practically relevant? Synergy, for example, is dose dependent, and that is nearly the most important part. How does that translate to the in vivo situation? So we were modeling synergy, which is a difficult concept by itself because you need to have a null hypothesis that analyzes that. So if synergy according to a model, which is subjective. So does that actually translate to the in vivo situation? So what do you see in patients, and in which types of cancers would that matter? And so the greater question is, of course, do simple endpoints, which we need to for AI to get enough data together, do they really help? So this was from a cancer cell line screen, but how do we go into the clinic? So we do get better numbers, but it doesn't matter. And so I have the impression sometimes you maybe only play a my numbers are higher than yours game in the end. Do we really advance the field? Well, that's not so clear in many cases, I think. And sometimes there's a third example. Sometimes you see what you want to see. So there are publications on using deep learning for electronic health records, for example. If you look into the abstract, you see deep learning achieves a high accuracy, and they report particular areas under the curve for in-hospital mortality and different other hospital-related endpoints. And so in different colors, you have different endpoints. And so these are the results from the deep learning model. And if you then go into the supplementary material, you have a logistic regression baseline model. And that baseline model basically achieves the same results. So the confidence intervals overlap in all except one case. Yeah. So that basically means that the abstract it promotes deep learning and says that is a very good result that this particular method achieves, but the baseline method is actually just as good. Okay. And if you do this type of bias reporting, that of course doesn't allow us to establish a realistic view on a particular field. Let's go back to biological data. So it's difficult to deal with, but is it impossible to deal with? Probably not. So where we can use, for example, gene expression data is in repurposing. And that, in my opinion, in our experience, but about 100 or so other publications say that as well. They actually say you can use gene expression data for repurposing reasonably well. And this is a setting where we have a one out of many selections. So it resembles more this early stage setting from before. So we don't need to make a prediction for a given molecule. We can see where we have a sufficient signal in order to pair a disease to a compound. And this is quite a good situation to be in because you have many options you can pick from. And so in our experience, transcriptomics data often contains sufficient signal for signal detection, but possibly less for modeling. So if you have noisy data, what you can do is you can look for extreme signals. That's what you can do for repurposing, but you don't need to understand that everything that happens in the middle, basically. And so with gene expression data, in our experience, quite often is like summarized here at the bottom. A third of the time, nothing happens. A third of the time, too much happens. So you cannot make sense of that. And a third of the time, you see something you can use. Though some of that you might have understood before in any way. And we can use it for really nice things in biology as well to test our compounds into a biological system. And this is one of my favorite studies by Yazaman, who finished her PhD 2 or 3 years ago. So we selected compounds to differentiate stem cells into cardiomyocytes. It takes 3 weeks. You can, on the genetic level and protein level, characterize your cells. They're relatively pure. And after 3 weeks, you really see a beating cardiomyocyte under the microscope. And so that really shows how what an impact chemistry and computer science can have to select chemical matter for biological setting. Yeah, a few slides of discussion at the end. So I would like to discuss the data we have right now, technical problems with model generation, and the relationship of AI and human beings, as well as AI and society. So what is the data we have right now? In my opinion, and I've encountered that many times, much of the data we generate, not all, but much at least, is generated for the wrong reasons or in wrong ways. So what do I mean by that? In many cases, we have proxy measures to reduce cost, but then the question is the data points we generate, how predictive are those for what we are actually interested in? And that can relate to cell systems, it can relate to animal studies, where in many cases, efficacy or safety related, predictivity is relatively low. In some cases, we have irrelevant systems, doses or time points. And one of the big problems right now is often we have hypothesis-free data. So basically, there's then the situation of here we have a pile of data. We didn't quite know why we generated it in this particular way, but maybe there's something we can find in it. And that's tricky, especially if you have biological systems. You have lots of noise, so you have lots of chance correlations. How do you find a signal if you have a signature of 20,000 genes? That is not trivial in many cases. The alternative, of course, is to do that in a hypothesis-driven way. So I label my protein of interest because I have some mechanistic hypothesis, and that is much cleaner data in many cases to deal with. That relates, of course, to the balance between having a technology push or a science pull. So in many cases, you might remember this example from the Human Genome Project. We have a new technology, so we're able to do something, generate new data, and this drives the data we generate. So it's not a science pull, where we have some kind of understanding at least in order to ask relevant questions, but we just generate data because we are able to. And that can be a tricky situation to then make sense of the data afterwards. And so this is a representation, how I summarized that. First comes the question, then the data, then the representation, and finally the method. And so first you have a question or hypothesis. So we identify key parameters or readouts that are needed for analysis, and they should be practically relevant. So we need to generate the right data. So we have our data, and they need to have a relevance, be of relevance for the question asked. So it needs to be the right data, it needs to be sufficient data, and it needs to be of sufficient quality. That involves also ontologies, for example, covering chemical space, whatever is relevant in a given question. So we have our underlying question or hypothesis, the data we generate, then we have a representation that captures the relevant information in the data. And finally, on top, we have the method. And of course, in some methods, if you have end-to-end learning, you can combine the top two parts. But method and representation always depend on data as well as question asked or hypothesis. And the key here is a method cannot save an unsuitable representation, and that cannot remedy an irrelevant question, an irrelevant, oops, sorry, I cannot even see what it is, cannot remedy irrelevant data for an ill thought through question. Okay, so there's a logical order in which we need to analyze our data. First comes the question that gives rise to the data we generate. Then we need to see what's the representation we need. And finally, we can apply a machine learning method. And the later stage in that pipeline cannot really remedy anything that went wrong before. So we need to think through those stages from the beginning. Quite often, if you have studies, there's the question, especially in complex settings such as drug discovery, is it really the method or is it by chance that happens? So if a drug comes out of the drug discovery pipeline, I mean, it's one of those concepts that don't really exist either, but if we have a drug that eventually gets approved, it's the result of a very long series of choices. So if we see claims in literature, AI discovers a drug against X, well, so even if it's a drug, so in the sense it's approved to the market at some stage, what is responsible? It's nearly impossible to say. So viewpoint A is we don't have a baseline for control, so we cannot really say whether it's the method. But of course, viewpoint B is, but it worked. Look, we have a compound that made it, in this case, to the market. And both are in some sense true at the same time, because we can in practice, we cannot have a comprehensive evaluation and continue development of 100 compounds in parallel to compare methods. So it's really difficult to do that. But the problem is, of course, that in the end, we have bias reporting, also in literature, for example. We don't have proper baseline controls. And sometimes what we also have is a focus on trivial wins. So of course, if you have a computational method you want to validate, you select compounds where you're more, where you believe that this compound really has a chance to have the desired effect. But in this manual way, we bias our model output. And in many cases, I think also the compounds that I eventually tested and so on, they are rather trivial modification of those that have been around before. So that doesn't really allow us to evaluate our methods properly. One conceptual problem, and I discussed it at length with Anthony Nichols from OpenAI, is how can we actually validate our models? And in some sense, there's only one way to really do that, and that's the large-scale, diverse, prospective validation. That is nearly impossible to do in practice. But the problem is if we have retrospective data sets, they are limited in size compared to all chemical space, and we don't really know the underlying distributions of all chemical space or the next chemical series we want to predict for. So we will never really be able to have a true estimate of model performance. And also if we have data sets, let's say the MUV, maximum unbiased validation data set, all those approaches only introduce another subjective bias. That still doesn't really allow us to say for a new chemical series that we're interested in, or for the next 1,000 new chemical series, how good will our model be? So there can only be large-scale prospective validation, but that is very, very difficult to do in practice. So the bigger picture, of course, AI is to a good part where it is due in no small part due to human psychology. So hype brings you money and fame, so realism is boring. There's also fear of missing out, of course. The others also do it, and beliefs often drive decisions. You have that on the personal level. You also have that on the organizational level, of course. Everyone needs a winner. So for example, after a company invested X million, you need to show success to justify the money that you have spent, which also leads to biases when reporting the outcome of particular methods. And selective reporting of successes then leads to everyone declaring victory, but in reality, no one knows what's actually going on and which methods work well and which ones don't. And it's difficult to really advance a field with little real comparison of methods. Okay, so to summarize, so we need to analyze our data as we did for many years before, absolutely. AI and deep learning are certainly a valuable tool in the toolbox. I think we can say that. The real game changer for AI and drug discovery will probably only come once we understand biology and biological data better, and when we are better able to generate it, so in a designed way, better experimental design, we know how to encode it and analyze it. Currently, there are a lot of computer science-driven approaches. Some are more applicable in drug discovery than others, but it's always important to really work on this interface. So if you have a bright idea from computer science, what is the right biological data that we need to input into the method? What do we need to generate if necessary to really answer the question? That is key. But that also translates to the need for better experimental design to actually generate data. And right now, there are many consortia, the IMI consortia, for example, that share data, but probably to really address those questions, we need consortia on an even larger scale, also for targeted data generation and not just sharing what is there already, because that might not fulfill the purpose that we need. Okay, thanks a lot for listening.
Thanks very much, Andreas. That was a tour de force. Can you hear me?
Yes.
Good. Okay, so I'd like to invite everyone who is present to ask questions. You should notice a Q&A button at the bottom of the Zoom interface where you can type your questions and they will pop up on my panel here, and then I'll pass them on to onto Andreas. So if anybody would like to think about questions, put them forward, just type them in. Please go ahead. So yeah, I thought that was a rousing talk, and it covered many aspects of drug discovery and the complexities of biology that we're fighting against in terms of developing our models. So do you think oh, we've got a question that's just come in. It's from Jose Brandao Neto, who I will, if I can just find you on the list, I can give you the microphone. So Jose, would you like to unmute yourself and ask your question?
Hi, can you hear me?
Yes, we can hear you. Go ahead.
So yeah, what's the best hypothesis-driven work you've seen recently?
Yeah, I think it's difficult to say the best. It's difficult to point that out. I think there's not one study I can point out here. I have to say many of the it depends on which area. Do you have a particular area you refer to? I think it's not really a question I can answer as such, I'm afraid.
Right. Okay. Yeah, I'm just
Sorry.
trying to see if you had one. Okay, cool. Thank you.
Thank you, Jose. I will mute you now. Next up is a question from Chris Swain, who I will see if I can pass the microphone to. Okay, Chris, can you hear us?
Yes, hopefully you can hear me as well.
Yes.
Yeah, go ahead, we can hear you.
Okay, you talk quite a bit about how complex biology is and how difficult it is to accommodate that, but there's also the concern that much of the early biology is possibly incorrect. So how do we accommodate that? Do we spend more effort going back, rechecking results?
It's a good question. There are studies from, I think, Bayer, for example, who found that 60% or whatever the number was of results were difficult to reproduce. I think what we could do possibly is correlate data from different sources, to see where do we find a consistent response, for example. If we have readouts on the gene expression level, different other levels, can we cross-correlate that? Because rechecking results, regenerating all data that is around is probably really tricky. Possibly to look for correlations in the data, check for plausibility. When we integrate data, maybe that's a practical way to do that.
Great.
Thanks.
Thanks, Chris. Okay, so we've got a question from Rajashi Guha. Rajashi, would you like to ask your question?
Sure, thanks. That was a really nice talk. What's your opinion of deep learning models applied to small data sets, sort of using methods like one-shot learning?
No.
Do you think endpoints such as those from secondary pharmacology are amenable to this type of approach?
Yeah, I think so. We do that in the group as well. So basically, it's secondary pharmacology data. You can also see if you have histopathology data and then human tox data. Yes, I think so. In our we have some preliminary results on that. I think that that makes sense. I think so. Yeah.
Cool, thank you.
Cool. Thanks, Rajashi. Okay, I don't see any remaining questions. I guess I was just going to ask one, which is you intimated that we need consortia who will generate data in a consistent and sensible way. Do you see any moves for that to happen in industry? Do you think that people will gather forces?
Yeah, so I discussed with people from different companies that people in also the big pharma companies, for example, see needs for that. It takes a while to assemble consortia. I mean, the IMI consortia is probably the biggest example of that. So it would be kind of the IMI on steroids, not just sharing data, but together generate relevant data that can be shared within consortia. I think you need a really big context for that. It could be the IMI context, for example, but it would need to be on a really big scale, I think, on a really high scale.
Okay, Benjamin Tahan, I'm not sure if I can, let me see if I'm trying to allow you to speak. So he has a question. Benjamin,
yeah, he doesn't have a speaker.
Yeah,
I can see the question here as well. Actually.
Yeah. Okay, so go ahead. So the question for everyone who doesn't see the question is, what are consortia such as Melody doing?
Yeah, Melody. So it's learning from data from distributed data sources. So basically in the large pharma companies, there's internal data, bioactivity data, for example, also very different types of data. And so the question is, how can you learn on local models and then pool the learning from those models into a joint model? So you don't share data, which is always a tricky aspect, of course, within pharma companies, but you're trying to jointly learn from data. Of course, then the data needs to be protected, so no one can hack into the data of another pharma company. But this is what Melody, for example, is doing. Currently exploring that for more early stage data, and at some stage later, it will then be for more complex data. That's the way I understand that.
Yeah, there's a similar initiative from LASA. I think it's called Kronos.
Yes.
Yeah.
Yeah, that's right. That's right.
Okay. Benjamin says thank you. Okay.
Thanks.
Oh, there's another question that's just come in from Sunil BVS. Let me just see if I can turn on your microphone. I see the microphone. Sunil, would you like to ask your question? Oh, similarly, they don't have a speaker. So the question was, any views on deep learning towards structure-based approaches using existing PDB knowledge?
Yeah, there was quite a lot happening in that area, of course. Yeah, machine learning, force fields, for example. I think there is a very good point in doing that. I was just examining a PhD student in Oxford earlier today. So there's a very good point in doing that. I think that's currently being explored. I think it's worthwhile doing. Of course, you need to see what the applicability domain of such models is, but I think that is worth doing. And there's lots going on, I have to say, right?
Mm-hmm. Okay, okay, this will be the last one, and then we have to move on to Vicky. So Olivier Stephen has said, let me see if I can get Olivier online. Olivier, would you like to ask your question? Olivier, would you like to ask your question? No, it seems like they're not going to. Okay, so they said, would you say that the AI applied in drug discovery will fade as rapidly as it came on the scene?
Well, I think there are things there that will stay. We have more data, and that may be it chemical data, biological data. Companies organize their data better. So we have proper ontologies. If you think about histopathology, if you think about other areas, I think there are things here that are here to stay. And I should say I'm very happy about that. But of course, there will be, if you follow this hype cycle, yeah, there might be a period where certain things might not survive. But of course, we will analyze data. I think it will not. It will not disappear. No. I don't think so. We will have better integrated data and new methods. Much of that, I hope, will survive.
Yeah, I think that's right. I think we'll reach a plateau of reality and understanding exactly how good they are and what their limitations are. Okay, good. So Andreas, thank you so much. That was a wonderful talk. I really do appreciate you coming to CompChem Kitchen to speak. If you'd like to stop sharing your screen, then Vicky will start sharing hers.
Sure, great. Thanks a lot.
Thank you. Vicky, welcome. Welcome to CompChem Kitchen.
Thank you. Is that working?
Yep.
Perfect.
Great. So Vicky is from Faculty 1000 Research, and she's going to tell us all about open access publishing and chem informatics. Take it away.
Perfect. Well, thank you for having me today. Yeah, so I'm going to give a little bit of a background on open access publishing in general anyway, and then talk a little bit about what we're doing at F1000 Research in terms of chemoinformatics publishing. So to begin with, I just kind of wanted to give a definition of what we actually mean by open science. It's a term bandied around a lot in recent years, and it's not always kind of clear what this means. So the idea is that I've kind of shared this quote where it's really kind of transparency throughout the process. So this can be open data, kind of open source in relation to software, but also kind of leading throughout the process. So open peer review and open access publishing as well. And really kind of the idea is if we embrace all the aspects of open science, then it will lead to some advantages that will kind of trickle down. So I've just listed them, some here on the right. So really the idea is if we embrace this kind of throughout the process, it will lead to more efficiency in kind of the gathering process of science, but also kind of further down as well. And this can lead to economic benefits and also really quality and integrity as well by having that transparency, and also kind of linking in the public kind of engagement as well. Often the public are the ones, the taxpayers are kind of funding the science that we actually do. So I think it's very important to kind of involve them in the process. So the open access movement really started in the kind of early 2000s with the Budapest Open Access Initiative. And then shortly afterwards, we had PubMed Central established, and also Biomed Central and PLOS were among some of the first kind of open access journals. And if you want to publish open access kind of today, there's various ways you can go about it, which I've listed on this slide. So the most popular and probably the most well-known currently is Gold Open Access. So this is really kind of publishing in an open access journal. The article is freely available at the point of publication. But most of the time with Gold journals, you're paying an article processing charge to make this work open and available for everyone, rather kind of than the traditional subscription journals where it's free to publish, but institutions are paying subscriptions to access these journals. We also have the hybrid option, which is traditional journals, subscription journals, which are behind paywalls, but you can pay a fee when you publish there to make your article in that journal open access. Hybrid's interesting as a lot of journals kind of defend this model and say this is a way where people can publish openly, but they still can publish in well-known titles. But actually a lot of funders are kind of critical of this model of open access, as it's kind of seen as double dipping. So particularly the Wellcome Trust are particularly critical. So the idea is that these hybrid journals are still getting revenue for charging subscriptions to institutions, but then are also making money on top where people are paying this fee to publish open access as well. Green is another form of open access, which is authors self-archiving, so on an institutional repository or something like that. It's completely free, but there are some kind of misconceptions around people aren't aware that they can do this. It's kind of a relatively straightforward thing to do, but what the journals often do is put an embargo on. So subscription journals will say, we've published your article, but you can't kind of self-archive it till 6 months or a year afterwards. So the idea behind this is obviously the journals kind of get first pick, but it does mean that it's delayed open access in that form. Preprints, relatively new to the kind of biological sciences and the chemical sciences, but they've been around in physics since the 90s with arXiv. So the idea kind of more recently, preprints have gained kind of more momentum and become more popular. And a lot of people feel this is kind of in response to the publishing process, which can often be very slow. A lot of people use preprints to kind of get feedback on their work. I think particularly bioRxiv, a lot of people envision that this could kind of replace the journal system entirely, but actually due to this kind of publish or perish culture where people are still rewarded on where they publish, still awarded funding and things like that, preprints are often used just kind of as an initial step in the process, and then people still pursue the kind of journal route later. So the advantages with open access, there's kind of a couple of them, but the main one that a lot of people kind of talk about is more citations and this citation advantage. There's kind of been various different studies on whether publishing open access does give you kind of more citations, and they haven't all been conclusive, but the majority of them do find that kind of publishing open access does give you more citations. Also, there's kind of broader societal impact as well. So having your work available to anyone with an internet connection means that you can kind of lead to more collaborations, or it can kind of be read by a wider readership. And not always, but often open access journals are interdisciplinary. So it can be a good way to get out work that doesn't always kind of neatly fit into a narrower scope of a traditional journal. But there are barriers to open access publishing as well. It's perhaps not as widespread as people envisioned it would be when the movement was initially set up. One of those barriers is actually publishers themselves, who are kind of often reluctant to make more of their portfolio open access, or kind of set up their old journals as new and flip them to open access journals. The publishers would say the kind of demand isn't there from researchers, which is true to some extent, and it's probably because of these two barriers here. So often unfairly, open access articles, journals are seen as not as high quality, or kind of have as much integrity as the traditional journals. And as we know, as I just mentioned, this can be tied to kind of promotions and funding, unfortunately. So this may be why open access journals aren't as popular. And also, there are other routes that are cost-free, as I just mentioned, but the most popular route is still gold open access, and article processing charges can really vary from journal to journal. Unfortunately, some of the journals that have higher impact factors have realized they can charge kind of higher article processing charges. So it can kind of range from lower prices up to, I think, around maybe £5,000 is one of the most expensive ones I've seen, which obviously is a lot to pay for publishing a paper, and particularly prohibitive to kind of researchers in the global south. So where we are with the current state of open access publishing, estimates say around kind of 28% of the scholarly literature is open access, but actually the discipline of chemistry and chemical engineering is one of the least likely ones to have articles kind of published open access. There is a lot of choice, so there are kind of over 13,000 open access journals listed in the directory of open access journals, so there's a lot to choose from. I think a sign of the times is the kind of American Chemical Society a couple of years ago set up their own open access journal. So kind of people are moving with these times and setting up kind of journals for the chemical sciences. And obviously I'll talk about how I think we're a good venue to publish in as well. And a growing number of research organisations and funders are having open access policies and mandates, which is been particularly interesting to kind of see this top-down approach. Plan S is a kind of European approach, although they're trying to recruit funders from kind of all over the world. And the idea is that this group of research funders are mandating that anything they fund needs to be published as open access from 2020, although it's been pushed back a bit now, so around 2021 onwards. In terms of kind of other open science initiatives that exist in chemistry, so the open databases allowing researchers access to relevant compound data, and then Chem Archive as well. The preprint server was set up in 2017, but it's been growing quite rapidly. And also around 30% of what's on Chem Archive at the moment is in the discipline of computational chemistry, which is exciting to see. So that's kind of the background on open access. Now on to a little bit about us. So we set up in 2013, so kind of relatively young for a journal in the scope of life sciences. More recently, we've kind of been looking at moving into the physical sciences a little bit more. And also we've been working with funders to help set them up with their own kind of open access publishing platforms as well. Really, when we kind of set up, we were really looking at the kind of traditional publishing process, which actually hasn't really changed that much in hundreds of years. And we wanted to do something a bit different and hopefully a kind of improved way of publishing research. So when we built the platform, we looked at kind of certain problems we wanted to tackle. So the first one really we were looking at was speed. So often kind of it's taking 6 months to a year to get a scientific article published. Often by that time it can be outdated, and obviously it's not ideal that it's kind of been stuck behind editorial closed doors at that point. So really we were inspired by the preprint servers, so we wanted to incorporate that into our publishing process, which I will go into more detail later. So we basically publish pretty much immediately, and then we do our peer review process post-publication. So this allows us to get kind of work out really quickly, removing any delay, and yeah, kind of having authors share on their own terms in that sense. We also felt there was this overemphasis on traditional research articles. We're not trying to get rid of traditional research articles. They're still the majority of what we're publishing, but we also wanted to be kind of flexible and allow researchers credit for the full range of research outputs they're producing. So things that don't always fit neatly into a traditional research article, perhaps they're not kind of a full story. So on the right here, you can see some examples of the different article types we publish. So from method articles to data research notes, which are basically kind of a 1 or 2 figure paper, opinion articles, systematic reviews, and also software tools as well, which we've actually found really popular on the platform since we've launched. Talking to a lot of kind of bioinformaticians, chemoinformaticians, software engineers, kind of finding that software often was ending up in the kind of method section of a traditional research article with the software engineer a middle author on that paper. What we really wanted to do with the software tool article was kind of give credit the software as its own citable item, and the researcher kind of full credit for the software they produced as well. So yeah, the idea behind this is kind of crediting for all outputs that researchers are producing, and also kind of reducing research waste as well. So allowing stuff to be published that, again, doesn't kind of fit that full story. We also really wanted to tackle what we saw as bias in the system. So particularly publication bias around kind of negative results, replication studies, and having this transparency throughout the process. So this starts with our authors. When they submit to us, we ask them to make their data suitably open. So deposit in a suitable repository, and kind of open to reviewers and readers as much as possible in line with the FAIR guidelines. And then we also have an open peer review process as well. So we publish review reports, and we also publish the reviewers' names as well. So it's a completely transparent process. So how does the publishing process actually work? So one of those article types I just went through is submitted to the platform and then checked over by our editorial team. Our editorial team really are just kind of doing sanity checks on the paper. So checking it's not plagiarized, methods adequately described, the author is from a registered affiliation, and kind of things like that. And the data is open, as I just mentioned, our team can kind of assist with that. If the paper kind of passes all those checks, it can typically be published within about 2 weeks. And at that point, it's published on the platform, it's an open access article, it's got a DOI, it's citable, but it's clearly labeled that it's awaiting peer review. We then start this peer review process post-publication. So we work with the authors who suggest suitable reviewers. We check them out for conflict of interest and kind of things like that. And then we invite the reviewers on the author's behalf, as a normal journal does. When the review reports come in, we give them a quick check over, but we publish them pretty much immediately. And we ask reviewers just to review the technical aspects of the work, not any perceived significance or novelty. So alongside their review reports, we also ask them to choose a status for the paper, which are these kind of 3 icons up here. So a green tick is an approved status. So it means the paper is kind of technically sound in all forms. Approved of reservations is like a major revision. And then the red cross is not approved. So that means there's some kind of fundamental flaws with how the work was carried out. So a paper needs to receive 2 ticks or 2 question marks and a tick to be indexed in PubMed. So those are our minimal kind of requirements for indexing. If the paper doesn't initially receive those statuses, then the authors can revise the paper, publish a new version, and then the referees can reassess the work. If they feel it's sufficiently improved, then they can update their statuses. So just to illustrate this process a bit more, as it is quite different to the traditional process, this is a paper we have published recently, which is awaiting the peer review process, as you can see here. And you can also see this is denoted in the square brackets behind the title. So this also forms part of the citation. So if anyone cites the work at this stage, it's clear it hasn't undergone peer review. And then this is a paper that has undergone peer review. So what you can see here are the reviewers' names on the right-hand side. We actually always encourage co-reviewing. We think it's a great way to kind of get early career researchers involved in the process. So you can see here, reviewer 1 chose to review with a colleague, and they've both been credited. And then you can see here that both reviewers have given the paper an approved of reservation status, which is updated in the square brackets behind the title. If you click read here, then you can read the peer review reports. I've just pulled them out on the next slide. So you can see what they look like. We've got the reviewer's name, affiliation, and all review reports also have their own DOI, so they are citable as well. We've got the status clearly labeled, the kind of the meat of the review report. And then what we ask authors to do when they're responding to referees is respond directly on the paper. So it's a bit more of an open dialogue process. We find it's kind of more reviewers and authors working together to improve the paper rather than kind of the anonymous process that exists. And then when the authors publish a new version of the paper, we ask them to fill in this amendments box, which makes it clear what has changed from the previous version. So you can see here, this is the same paper with the version 2 published. So you actually can access all the previous versions of the paper. They're individually citable, so you can see how the paper's changed. You can see here with the version 2 that both referees reviewed the paper, felt it had improved sufficiently, and updated their statuses to a tick. So the paper would have been indexed in PubMed at this point. The nice thing about this versioning system as well is that we actually do allow small updates at any stage as well. So it can be particularly useful for kind of method articles or software tool articles where if you've kind of tweaked something 6 months later, it's not enough to kind of publish a whole new paper, but you want to keep the old one cohesive, then you can publish small updates at any point as well. So now I wanted to talk to you about F1000 research gateways and the Chemical Information Science Gateway. So gateways are part of the platform where they're kind of customizable hubs for kind of communities, organizations, or they can be thematic like the Chemical Information Science one. And we kind of almost think of them as mini journals within the platform. The publishing process works exactly the same way, but they're kind of got their branding on as well. So this is what our Chemical Information Science Gateway looks like. This is the landing page here. And really, this was actually one of our first gateways that we launched in 2015 and really kind of spearheaded by Jürgen here, who's on our advisory board. He kind of foresaw it as a way to kind of bridge the gap in chemistry and open access, and kind of wanted a flagship journal he felt that didn't exist in this kind of discipline, but incorporated kind of fully openness, but also kind of give an opportunity for unconventional submissions as well. So this is kind of our browse page with the articles on it. So the current scope we've got is kind of all computational approaches to kind of represent and analyze chemical structures and chemical data. AI, machine learning, all welcome. And also kind of any contributions reporting negative data or kind of showing why computational methods fail as well. Papers are kind of initially checked for suitability by the advisory board, and then we have the normal publication process, which I just went through. So we've actually published 20 papers so far, which, to be frank, we are kind of disappointed with the uptake. Me and Jürgen have had many discussions on kind of why this could be. One of the kind of conclusions we thought was perhaps it was almost a bit ahead of its time in 2015. Chem Archive, the preprint server, wasn't even set up till 2017. So perhaps kind of open peer review and this full open approach, we perhaps kind of jumped the gun too soon. It'd actually be really interesting to get your thoughts. I know there are kind of other chemoinformatics journals as well, but I feel like there are a lot of advantages to publish here. So yeah, it would be good to kind of get your thoughts after this. But what we have published has been a kind of a mix of all the different article types we support, which I think is actually really great to see and shows it's kind of useful for a range of outputs. And again, what we have published has really been highly viewed and cited as well. So it's good to know that the work that's being published in this gateway is having an impact. We're also currently running a joint call for papers as well with our Python collection. So if you do have any kind of Python software that's kind of chemoinformatics related, then definitely consider submitting as part of this call. We're going to be featuring papers in a blog and kind of do some social media promotion later this year as well. And finally, I just kind of wanted to pull out again just some advantages we feel kind of publishing on the CIS gateway. So I would say kind of speed. I hope I've made it clear it's a very kind of quick process, inclusive from the range of different research outputs you can publish here. And we do really want to kind of keep it community-based as well. So we did refresh the advisory board this year to kind of get some more early career researchers on it. And also with that open peer review process as well, we feel it's kind of ideal to be kind of working with the community. Our APCs are pretty cheap compared to other open access journals. So we actually do our APCs by word count. So that can range from $150 to $1,000. Most of the software tools we publish come out as Medium articles. So that's around $500. So definitely kind of, yeah, cheaper than some other journals. And also we kind of feel as this gateway grows, which we hope it will, and we continue to promote it, that it will be kind of visibility as being part of this, and we will continue to kind of promote it as well. And then lastly, I just wanted to show off that we're trying to bring more kind of interactivity into our articles. We think a lot of journals still publish as if papers are being published in paper format. So for our HTML version of our articles, we really wanted to kind of bring more stuff in. So this is an interactive figure that we can host, which was created in Plotly. If you're not using Plotly, then we can also use an iframe to kind of bring in anything else you've created. So I think this is the Shiny app here. And then also we have Code Ocean widgets that can be part of an article as well. So the kind of code's all in here for anyone accessing the article to play around with, run reanalysis. So it's basically a Docker, so you can kind of run all the code within the article without having to navigate off it. Cool, thank you very much.
Great, thank you very much. Thank you, that was great. Good, so if anybody would like to ask questions, please type away. Set that up. Okay, good. So Christopher Southern has raised his hand. So let me see if I can, Christopher, there we go. Okay, Christopher, would you like to ask your question? Hello, Chris, can you hear us?
Yeah, try again. How's that?
Yeah.
I can hear you.
Yes,
Great. Okay.
yeah.
So for this, for the gateway, how explicit is the chemistry? Do you have machine extractable structures, smile strings, inches, or PubChem IDs in the articles?
I don't think we do actually. That is actually a really good point. I think for us, because we were always, our background's kind of been life science focused, perhaps we kind of have missed some stuff, but that would be good actually. Yeah, I'm going to take a note of that to have a look because.
Yeah, I can contact you, yeah.
Yeah, that would be great actually. I think I left my email on the last slide. But
good, fine, yeah, okay.
Yeah, that would be great. That would be a good thing for us to be looking at actually.
Yeah, you can also send chats directly to any participant, Chris.
Okay, great.
Okay, good. So we've now got, I'm gonna turn off your, take the microphone back from you, Chris. So next up is a question from Sang. No. So Sang, would you like to ask your question?
Yeah, hello, can you hear me?
Yes, you can hear me. Go ahead.
Yeah, hi. So you said that there's a lot of Python code support for when you publish articles in this. So in my previous experience of having like code sections in papers and stuff like that, I always find that the versions they have is often outdated. For example, Python. So Python, for example, have a Python 2.7 versus like Python 3.8, which is like the standard version now. So I was wondering, like, do you notify the editor, the publishers, like this code is outdated and you need to update the code, or is it like, is that something just, is it not, is it, oh, I mean, so how does the code support work and the library, like backend support work or something like that? Yeah.
Yeah.
So if that makes sense.
Yeah, no, it does make sense. So basically, oh no, have you got anything else?
No, that's right.
Okay, cool, sorry. I thought I cut you off there. Yeah, so basically, what we ask authors to do when they submit to us is we're happy for them to link to GitHub. That's completely fine. And we can have that in the data availability section in the article. But we also ask them to put an archived version of the code at Zenodo, just so it's we kind of have a permanent record when the article is published with us. So in terms of us kind of notifying authors that there's kind of been a new one, we don't do anything currently. The onus is kind of currently on the authors to come to us and update their articles, but I think ideally a point we're kind of exploring at, and we do want to get to, is that we'd like it to be a kind of automated system. So we do some work with Cytoscape, who create their own apps, and then they kind of have versioning systems with that as well. So what we'd like to do is that if someone's published a paper on an app, and then they update that app, then it kind of pulls through to the article as well. So that's not something we currently have, but we definitely kind of want to explore that at some point to kind of save someone updating every kind of record of where it is and make it a bit more seamless.
Okay, cool.
Cool. Thank you, Sang. So yeah, I'm curious about if you can host things like IPython notebooks.
We've been looking into hosting Jupyter notebooks, which is definitely something we're kind of looking at this year. It's just, for us, it's kind of working out how much uptake there would be. Currently with the iframes, where it just kind of pulls through from where it's hosted elsewhere, the reason we do that is really kind of just security. We have hosted kind of living data before, but it meant one of our production editors literally kind of sitting through and checking there was no security risks and stuff we were hosting ourselves. So I think with the notebooks, we might run into some issues around that, but yeah, we are looking into that.
Mm-hmm. Cool. Great. So if, we just had a question from, comment from Antonina. She says, doesn't have a question. However, the Faculty 1000 talk was extremely informative and educational. Learned a lot. Cheers.
I'm glad, thanks. Sometimes when it's a comment, not a question, you get annoyed, but that one was nice.
Yeah, cool, thanks. Good, yeah, I thought that was very, very helpful.
Perfect, yeah, definitely get in touch if you've kind of got any ideas, anyone, of how we can kind of bring more of you guys into it. Like I said, it's kind of our only kind of chemistry thing that we've got on the site, so we definitely kind of need to learn a bit more from the community.
Yeah, F1000 research looks like a really, really interesting venue, especially if you can publish things like data sets and even negative results, which are really useful, hard to get published. So
yeah, definitely.
Great.
Cool.
So unless there are any more questions, I'm trying to think if there are any announcements. There is still a job posted for the Oxford Protein Informatics group, so check out the blowpig.com, blopig.com website for details about that. The Molecular Graphics and Modeling Society lecture tour, unfortunately, has been postponed indefinitely. So watch the mgms.org website for that. I'm trying to think if there's anything else that we need to mention. I think that's pretty much it. I'm sure.
Well, just
one more.
Just to interrupt you, Garrett, and to say to everybody here, if they've got suggestions for a similar event that we could run next term, then of course to everybody to email us, and
absolutely,
and your suggestions are most welcome, right? So please do email us with your suggestions. And thanks, my own thanks to Vicky and Andreas for 2 great talks for this evening. Thank you very much indeed. That was absolutely fabulous. Thank you.
Thank you.
We've just had a question come in from Dominic Schwartz, who, let me see if I can give you the microphone. Dominic, would you like to go ahead?
Can you hear me?
Yes, we can hear you. Go ahead. Okay,
nice. Hi, I just wondered if there has been an analysis, not necessarily by you, but like in general, that you know of, how the rejection rates change when you have a blind review process versus an open review process where the reviewers' names are known.
I think it's an interesting one. I think there probably is some literature out there. I think we're a bit hard to include in that because we don't necessarily kind of have reject per se. So papers can get not approved statuses, but they do get not approved statuses, they remain on the platform and the authors are still able to kind of revise and put more work in to improve the paper. So we kind of don't have rejection per se, but I think open peer review is really interesting. I definitely don't think we have enough data on it. I think research on research is actually very kind of underfunded. I think our own experiences with open peer review is that people are always worried it won't be as critical as blind peer review, but we haven't found that. We've just found that when people are critical, I think they're more constructive than perhaps they are if they're kind of anonymous. So we have had people be very critical of papers, but yeah, kind of offer suggestions on how to help it. But yeah, the BMC have a research integrity journal, which does have a lot of that kind of content on there. But yeah, it's definitely a really interesting kind of field of looking into research. And I definitely think we need more on it.
Thanks.
Cool. Thanks, Dominic. So we also just had a thank you from Sunil BVS, who says, thanks to CCK organizers and speakers for organizing insightful talks. We've joined from India and looking forward to more virtual CCKs in future. So yay, we're global for this platform. So I really do hope that we get to organize more of these. It's been a great, I think, a great experience. Wendy War has just commented, and Wendy, I'll see if I can give you the microphone.
He's also on our advisory board as well for the Gateway.
Okay. Wendy, would you like to go ahead? It looks like Wendy's not unmuted. Wendy said JAMA runs a prestigious conference on peer review. Let's see. Okay, is somebody? Okay, good. Yeah, and someone else has just commented. Thank you so much to CCK. Thank you so much, CCK-18, for very interesting lecture series. It's very useful. So good, great. So, oh gosh, more thanks coming in. Jana Sara said thank you. Good. All right, I think we should probably call it an evening. And thank you very much to both Andreas Bender from the University of Cambridge and AstraZeneca, and Vicky Hellon from F1000 Research for really, really great talks. Really enjoyed them. And these talks will be made available online afterwards. And we have been streaming live as well on YouTube. So thanks everyone for taking part. Just stopping